Our next speaker is also a researcher from ICMAB. She's Aida Carreño. She's a biotechnologist and doctor in material science. And she works in the development of nanovesicles toward directed administration of enzymes. And she will talk about techniques for separation and quantification of biomolecules.
Okay, hello everybody. So today I wanted to talk about some techniques that are used for separation and quantification of biomolecules. In this talk, I will start with a brief introduction of what are biomolecules and say some examples of applications that they can have in nanomedicine. Also, I want to have an overview of the conventional methods that have been usually used for the quantification of pure biomolecules and also other alternative and optimization of these ones. I also want to do a brief highlight about methodologies such as enzyme-linked immunoabsorbent assay, also named ELISA, for the analysis of complex samples. And finally, I want to talk about alternative separation techniques that were initially were developed for the purification of biomolecules from complex samples, but they can also be used for the quantification of these biomolecules. Both analytical techniques that I want to explain here will be the high-pressure liquid chromatography and also the gel electrophoresis. And finally, I will end with a particular case of a liposomal-based formulation in which some of these methodologies that I will explain during the talk are being used routinely during the manufacturing of this formulation. So first of all, today here we are all talking about biomolecules, but what are biomolecules? So biomolecules are macromolecules that are present in living organisms. They are a huge variety of them, but they can be mainly classified as lipids, carbohydrates, nucleic acids, and proteins. These complex structures have a function in the living organism, but they are not easily implemented in medicine in an individual way because they have some limitations like high immunogenicity, low cell permeability, and high instability. So as all of us know, during last decades, nanotechnology allowed their combination in different nanomaterials in order to be capable to apply these particular biomolecules in medicine applications such as targeted therapy, also in vaccines for prevention, bioimaging for monitoring of nanoparticles and also of some diseases like tumor, but they are also starting to be implemented in medical devices. So as they are the objective of their use is for medicine, it's important to have a good quality control of these materials in order to ensure the batch-to-batch reproducibility of them, but also the efficacy and safety previous to come to preclinical or clinical tests with them. And the regulatory agencies, like the European Medicine Agency, recommends to identify the critical quality attributes that are those features of these nanoparticles that can have a high impact on their physicochemical properties. Some of them are described here, but today I wanted to have a highlight in the importance on determining the chemical composition and the drug entrapment efficiency that we have in different nanomaterials, because we need to know exactly what we have in our formulations when we do dosage of them in vivo. So for the quantification of these biomolecules, the most known conventional method is the use of the spectrophotometry. This method is based on UV absorbance and the concentration of our molecule can be calculated using the Beer-Lambert equation that correlate the concentration of this biomolecule with the absorbance units obtained and the coefficient extinction of the biomolecule, and also the light path of the spectrophotometric cuvette used for the analysis. In particular, for the analysis of nucleic acids and proteins, their absorbance, or their major signal in the absorbance, is when we analyze them in the wavelength absorbance of 260 nanometers for nucleic acids and 280 for protein. And this method is a fast and simple method, so that doesn't require any sample preparation, but it have some disadvantages, because when we have biomolecules that absorb in near wavelength, like in these examples, we can have some interference if our sample is contaminated. However, this also can be a benefit because they are described in literature, the different purity ratios that we can obtain from the ratio of these absorbance, and we can have an idea of the percentage of protein or nucleic acid that we have in our sample. Also, it's interesting to know that with this methodology, we can analyze our protein samples, for example, at the absorbance of 320, because at this absorbance, if we have values lower than 0.02, we will know that there are no dispersion in the sample, and that will mean that our protein have no aggregates there. However, as we are working with nanoparticles, all of us know that nanoparticles have a specific size and they can disperse light. So this methodology, it's not commonly it's not a useful method to study the quantification of our biomolecules entrapped in the enzymes. There are other alternative methods to quantify, for example, proteins that are indirect methods that consist in colorimetric or fluorescence assays. These assays, that some examples could be the Lowry, Bradford, or BCA assays, used specific dyes that are bound on the amino acid of the proteins, and using a commercial standard curve of bovine serum albumin, we can extrapolate the concentration that we have in our samples. However, these methods have different disadvantages. The one is that if we have a mixture of different proteins, we could not discriminate by the one that is of our interest. We will detect all the protein content in our samples. Is higher time consuming than the spectrophotometry because we need to do serial dilutions of the sample, and we have to wait for the interaction between dye and our sample to have the colorimetric reaction. And also, in most of the cases, there are some interference related to the presence of the lipids or detergents or reducing agents that can also have can impact in the colorimetric assay and have overestimated results. These assays are being optimized in these last years for fluorescence assays that have higher sensitivity. We can achieve the determination of concentration of picograms per milliliter. However, most of them are still incompatible with detergents, and in most of our nanoparticles, we used some detergent as excipient to stabilize the particles. So, in order to have an alternative, I wanted to talk about the ELISA assay that can be used for complex samples. In specific, is normally used for the detection of macromolecules when we have our biomolecules coming from serum that we extract for in vivo experiments, or from the cell lysis that we used for the in vitro studies. This assay is based on the immobilization of the target molecule that we want to quantify in a solid surface, and then is binding. We need a binding a specific antibody in order to specifically binding our targeted molecule and only get signals from them. There are different formats of ELISA. There are the direct ELISA, indirect ELISA, and sandwich ELISA. All of them are based in the same assay basis, but the difference is the number of the antibodies that we have in the kit, in the commercial kit. The one is the direct ELISA, where we only have one specific antibody, one primary antibody that will interact with our molecule of interest, and this one have the reporter enzyme, fluorophore, or chemiluminescent fluorescence in order to give the signal. This one is the cheaper because in the kit there is only one antibody, and we all know that biomolecules are expensive, but it's true that it's the less sensitive because we can have non-specific interactions in the surface. So the most used is the indirect ELISA, where we have a primary antibody that have this specific binding to the targeting molecule, but then we have also a secondary one that is specific to this primary antibody, and this one have the reporter signal. In this way, during these bindings, we have a first binding step with this antibody, then we have to clean the excess of this antibody, and we perform the secondary antibody binding. In this way, the signal is more specific. Also, we can go to the sandwich ELISA, where in the plate, normally these kits are done with multi-plate, with 96 well plates. In the plate, we have already a specific antibody for the macromolecule that we want to analyze, and then we have, again, two different antibodies for the analysis. This kind of kit is very interesting and have high specificity. A lot of samples can be analyzed in a same well plate because we have 96 wells. However, it's highly time consuming because we need a lot of time of incubation and cleaning, and also they are very expensive kits due to that we are working with recombinant proteins. So, as alternative to all of these techniques, today I wanted to do a little overview of different the high pressure liquid chromatography, HPLC analysis, and gene electrophoresis, that are two methodologies that are well implemented at a research level in laboratories. And they mainly are used for the separation of biomolecules in complex samples, but also can be used for the quantification of these biomolecules. So first, I wanted to start with the HPLC analysis for the chemical composition of the total content of our nanoparticles. So this methodology is based on the separation of different molecules regarding a specific physicochemical property that can be size, charge, hydrophobicity, or other specific interactions. This separation will be defined by the combination of a stationary phase and a mobile phase. The stationary phase is the solid phase where our molecule will be retained. This one is found in the resin of the HPLC columns, and depending on this nature, the interaction will be regarding the different physicochemical properties of our molecule. The mobile phase instead will be liquid phase that we will prepare, and normally there are some ones that are defined from specific HPLC columns and chromatographies, and we will use them, we will flow them through the stationary phase in order to allow the retention or the separation of the molecule in the different time of the process. This flux of the liquid phase can be used as an isocratic mode, that it will mean that the same composition of the mobile phase will be flowing through the column during all the analysis, or by gradient mode. That will mean that we will start with a mobile phase 1A, for example, that will have a specific composition, and then this one will be useful for the retention of the molecule, and then we will use the mobile phase B, or a mixture of more of one, in order to achieve the elution of the retention molecules. So as a general characteristics, this is an example of an HPLC equipment. Mostly, we have to have an idea that an HPLC equipment is mostly a pump that is used for the flow of the mobile phases that have different modules. The principal one is this, the pump, but then also have an injector where we will put our samples in specific vials in order to the equipment to take a known volume of sample and then inject to the system to be capable to reach the column phase that will be put in the column compartment. This compartment will allow us also the control of the temperature, because in determinate chromatographies, we will need to control this temperature in order to avoid aggregation or in order to modify the solubility of our molecules. Then, after the chromatography step or the separation step, the step of the molecules inside this stationary phase, this compartment is connected to a detector. And then here, I want to say that this methodology have a really high versatility because there exist a lot of types of detectors. The most used is the UV or the other array detector that is based on the UV visible absorbance. And this is the more commonly used in the pharma quality control. However, there are some specific chemical compounds that cannot be observed by the absorbance. They didn't absorb at this wavelength range. And then there are alternatives like the evaporative light scattering detector that is based on the light scattering. It is also interesting to say that these equipments can also be coupled to mass spectrometry in order to analyze the molecular weight of our samples. And also this is a great opportunity, as said, to be capable to have another approach that it could be related with the identification of the compound in case that we didn't know the molecular structure of this. And depending on specific applications, there are other detectors also that are based on refractive index, fluorescence, or conductivity. It's interesting to say that we are not limited to use only one detector, if not that we can use several of them in tandem, and then we can analyze our samples or detect them by different bases. One of the also important things to have in mind when we work with this kind of techniques, that it's very important to be aware on the sample preparation, because all these techniques were first developed for individual materials, but now here we are working with nanomaterials, and if we didn't prepare a proper preparation, we can lose some material during this step. And this is important because if we have a nanoparticle dispersion, if we didn't disrupt them with these techniques, we need to filtrate both the samples but also the mobile phases before to introduce them in the equipment in order to avoid overpressure issues. So if we have the nanoparticles not well solubilized, we could lose some material during this preparation. So very briefly, there exist different liquid chromatography types, and as I said, that depends on the stationary phase. Here I put the most relevant ones. The first one is the ion exchange chromatography that is based on the separation of molecules by charge. Here we need to choose very careful the mobile phase in order to have a controlled pH, to have the charge of the molecule necessary for use with our columns. There are two types of columns. The cationic ones are negative, have negatively charged resins groups in the resin, so they are useful for the capture or the retention of positive molecules. And in contrast, the anionic ones are the contrary, and they are used for the retention of negative molecules. This chromatography is normally used for biotechnology applications like the purification of proteins. Then there is also the option of the size exclusion chromatography, that these columns used porous resins that allow the separation of the molecules by molecular size. Normally in proteins, the common buffer used is the phosphate buffer saline, and the application of this chromatography is principally the detection of the molecular weight of your sample, of your biomolecule, also the characterization of the aggregation percentage of your samples, but also it can be used for release studies applications where we can have the nanoparticle and the released drug, and then the released drug should be with lower size, so it will be more retaining inside the porus of the silica, and then the nanoparticles with higher size will be firstly eluted. In this chromatography, it's important to have in account that we have a limited sample volume to work, because if we surpass this volume, we will collimate the chromatography and we will not have a good separation of the samples. Also, there are other chromatographies based on affinity. There are different types. There can be the most commonly used are immobilized metal affinity chromatography that can have metal ion-based metal ions in resins such as nickel and cobalt. And for example, these ones are also useful for the purification of proteins, since the histidine groups can interact with these metal ions. And then for the elution, we need to put imidazole in the elution mobile phases. However, for specific applications, we also can buy specific columns with specific antibodies in order, or other molecules, in order to allow the specific binding of our sample to the column, and then knowing the release method or the breakage base of this interaction, we could elute our particles. Finally, the most used chromatography for quantitative analysis in the industry is the reverse phase, which is based on the hydrophobic interactions between the stationary phase and our molecule. Here, the common polar solvents are mobile phase based in water, or others organic solvents like methanol and acetonitride. These columns are based on resins that have long hydrocarbon chains, and they are named according to the name of carbon chains carbon numbers that they have, C4, C8, or C18. There are several ones. Okay, so as a take-home message of this methodology, we could say that it's a methodology that have a rapid analysis, but also accurate and reproducible, and have and give quantitative analytical results. However, this method requires a complex operation and high technical skill, and for this reason, the development of the method is quite extensive in time. It's quite time consuming, but then once you have the method, this equipment could be used as a high throughput analysis also. It is also interesting the high resolution and sensitivity of this method, that we can achieve the analysis of microliters per milliliters proteins, and also the fact that I said before that we have the versatility of use different detection modes. Okay. On the other hand, I also want to take of gel electrophoresis methods that are commonly used in genetics, microbiology, and other biology labs that are used for the detection of different biomolecules, or as nucleic acid or proteins. These methods are based on the visualization and separation of these nucleic acids or proteins based on their molecular size. And they are based through a porous gel by applying an electric field. There are different operation modes also of this gel electrophoresis, but we have to take in account that when we want to analyze nucleic acids, we will use gels of nature made of agarose, and when we want to analyze proteins, we will use acrylamide gels. So there are, in all cases, we need to have a standard mixture of DNA or proteins of known molecular weight in order to analyze our protein and determine the molecular weight. But in the case, in conventional methods, we also need staining requirements in both cases. But for the acrylamide gels, now Bio-Rad have a stain-free acrylamide that is based on trisglycine extended, then mark fluorescently the tryptophans of the proteins, and then this label is not necessary. Also, an applicability of this is that from both electrophoretic gels, we can cut the band of interest and then send to the genomic center to sequence the nucleic acid sequence that we want to know, or we also can send the protein to a mass spectrometer service. To final this part, when we want to also identify, when we have a complex mixture of nucleic acids or proteins, and we want to identify if our molecule of interest is there, we can use blotting methods that, in the case of agarose gels, are called Southerborn or Northernborn, depending the nucleic acid type that we are blotting, and in the acrylamide gels are the Western blot. In general, these procedures are based on the transferring of these electrophoretic bands from the gel from another membrane, and then after the hybridization of complement DNA radiolabeled or with marked antibodies, we can have a specific signal of this. So this method have also high sensitivity, has lower cost than the HPLC method, except when we use this TGX acrylamide because it's very expensive, or when we work with the blotting material because there we use antibodies. The good part is that we have standardized rapid sample preparation by the use of a specific buffer that is called Laembli. And then they also can be used for the detection of impurity profiles, because if we have some degradation in the nucleic acid or the protein, we will observe several bands in in the gel. However, again, if we never work with this, the operation is complex and the technical skills are required. There are poor gel-to-gel reproducibility and low accuracy. So this method is established as semi-quantitative method because the software has some limitations since they analyze the densitometry of the bands. And when these are saturated, the response is not so much linear. And also that the samples to charge in a well, in a gel, is limited by the number of wells of this. So very quickly, I have 2 minutes. I want to talk about a specific case that I will pass this. It's a liposomal-based formulation that has been developed here in the nanomol bio group. This formulation has entrapped an enzyme that is the active substance. And during the manufacturing step of this, we implemented the use of different techniques that I explained here. For example, during the synthesis of the API, only the enzyme, we have this procedure step where we produced our recombinant protein in mammalian cells, but then we used a combination of chromatographic techniques in order to purify the enzyme from the cell lysase that we obtain from this media. Also, before to entrap this in the liposomes, our partners, Limbio, made the certificate of analysis by characterizing different physicochemical properties of this enzyme, such as the content by absorbance, since it's a poor enzyme, but also they use the Western blot to identify if the protein is there and it's well produced. And also they used both electrophoresis and site exclusion chromatography in order to know the purity in terms of aggregates and also the molecular weight of the sample in order to see if the sequence has been well produced or it has some loss of amino acids. And also after this, our partners, Nanomol Technologies, produced these liposomal-based formulations entrapping the enzyme, purify them by tangential flow filtration in order to remove all the free GLA and sterilize them. And we analyzed the most critical parameters is the analysis of chemical composition in terms of lipid content of this. We developed a reverse phase method where we have an extensive time-consuming of development because of the need of optimization of sample preparation, also standard cube and others. But we were able to detect different lipid and surfactant molecules. And then in the initial steps of this analysis, of development of this formulation, we use this TGX acrylamide gel electrophoresis for the quantification or the semi-quantification of the protein content in these samples using a standard cure. But since this is not very accurate, we also, with the help of our Limbio partner, developed another different reverse HPLC method in order to do this specific quantification also by HPLC. And this is all. Thank you.
Thank you very much, Aida. Is there any question from the audience?
Yeah, thank you for the nice talk, super nice. I was just wondering regarding the HPLC method, when we use them with different stationary phases, the columns, is there any restrictions in the kind of columns we can use depending with nanomaterials, I mean? Depending on nanoparticle, I don't know, size, composition. I was just a bit more worried about size, maybe, that they can get stuck in the columns. So I don't know how do you manage how to use or which one to use?
Yeah, in our case, we have lipid vesicles, and we were capable to disrupt. But in the case to have methyl or other particles, this would be not possible. So you should be aware on the particle size, on the porosity of the column. There are different versions on size of the resin, longitude also of the column, and depending on this, you could achieve different the chromatography of different sized nanomaterials.
Okay.
But also, my recommendation is if you are thinking to buy something, for me, I always write to Agilent or Waters, that are the most common commercial houses of columns, and they are more expert in this.
Oh, okay, okay.
Yeah, for example, for the reverse phase, they told to me that for the analysis of big biomolecules, such proteins, it's better to use the C4 columns than the C18.
18,
Yeah, yeah. Also by the molecular weight of the molecule.
Okay, I was asking because we usually use size exclusion, just you are safe in that range, no? Usually if you do size exclusion chromatography. But it would be also nice to use other kind of stationary phases, and yeah, I was just curious. Thank you.
Thank you, Aida. Sorry, one question regarding ELISA. I know your talk was not focused on ELISA, but since you talked at the beginning, since it's a colorimetric method, do you have limitations with the color of your original sample? I mean, can you work with colored solutions?
Can be colorimetric, but also fluorimetric or chemiluminescence based. So, and the ELISA kit could have different detection modes regarding the reporter molecule that you have in the antibody. If you have an enzyme, normally there are the colorimetric reaction, and then the results will be colorimetric, but you also can have dyes on other chemiluminescent substrates.
Okay, thank you.
Yeah.
Any other question? I was wondering about HPLC. You mentioned that you can have different types of detectors. How modular is that? I mean, you can have an HPLC equipment and you have like X detectors and you interchange them, or is something that it's more fixed? Because it's very important for the detection of your analyte, right?
Yeah, but you can interchange them as you want.
It's very modular.
Yeah.
It's not that you
Yeah.
have to go to look for an HPLC with this kind of detector to...
No, the most commonly used is the UV, because also is the most cheaper, because each module is very expensive. And then, for example, here in the ICMAB, we have both. We have the DAD array that it's UV based, absorbance based, and we also have the evaporative light scattering, that I use this one for my formulation because I have a phospholipid that cannot absorb in the UV range. But there are others. For example, in the survey of the analysis of chemica here on the Autónoma, there is also the option of the HPLC mass spectra. So you can go there if you need to analyze the molecular size of your particles in order to do the experiments. But these detectors are not so common because they are very, very expensive.